Hello everyone, I am Efsun Güler and my teammates of this project are İdil, Ataç and Egekor. Today we are going to present our EEE 473 term project, which is called as MRI simulator with rectilinear scanning, considering effects of B0 inhomogeneity and chemical shift as non-idealities. First of all, the effects of chemical shift and magnetic field inhomogeneities were imposed on the original MRI image. Then, the image was reconstructed using rectilinear scanning trajectory. In class, we have analyzed the imaging equations of MRI, but we didn't fulfill the mathematical derivations for imaging equations when B0 inhomogeneity and chemical shift are present. The reconstruction of the image was performed by using inverse fast Fourier transform algorithms of MATLAB, and this means the acquisition of an MRI image from a slice of the body. Our first assumption is that the magnetic field in the device deviates from its ideal value circularly from its center to its peripherals. And the second assumption was to consider fatty tissues, such as white matter and subdermal materials, as agents of chemical shift in the reconstructed image. For the methods part of our project, as a realistic parameter, B0 inhomogeneity is the first non-ideality considered in our project. As given on the slide, the formula showing the B0 inhomogeneity depends on the magnetic field and the diamagnetic susceptibility. Magnetic susceptibility is a material property being responsible for decreasing or increasing the magnetic field within the material with respect to the surrounding field. Another realistic parameter included in our MRI simulator is known as chemical shift. It measures the change in Larmor frequency because of the chemical environment of the image nucleus. This charge varies from tissue to tissue. As you can see on the formula given on the slide, V corresponds to the Larmor frequency and the symbol corresponds to the shielding constant. In our project, we analyzed the chemical shift caused by the fatty tissues, subdermal tissues around the skull, and the myelin sheets of the white matter compose the fat tissues located in the human body in an MRI scanner. It was assumed that the shielding constants of these tissues is 3.35 ppm throughout the project.
In order to construct the case-space representation of the image, we first sampled the image with the sampling time Ts, where nx is the number of desired pixels and the t is the unit duration between the sampled points. Here the unit duration is related to nx, full width half maximum, gyromagnetic ratio, and the gradient magnetic field. For this purpose, full width half maximum is chosen according to the desired final resolution, and the other values were taken as the constants, which will be shown later. While reconstructing the image, first we have worked on the ideal case. In order to construct the case-space representation, we have used the discrete Fourier transform shown in the slide. The image is scanned rectilinearly, and the samples were used as magnitudes of the 2D complex exponentials. We scanned all points and added the exponentials on top of each other in a 2D array. Then the images were reconstructed in the space domain using inverse fast Fourier transform. Then we focused on the effect of B0 inhomogeneity in the reconstructed image. It is assumed that the magnetic field on the device deviates from its original value from the center towards its peripherals. The increase in the magnetic field causes an upward shift in the frequencies of the complex exponentials of the effective voxels correlated with the acquisition time of this point. The exponential which corresponds to these effects is multiplied with the ideal exponential in order to resemble the B0 inhomogeneity effect. Upon adding this effect, the image was reconstructed as in the ideal case. In the case of chemical shift, it is acknowledged that the fatty tissues cause chemical shift. The image is masked to indicate the locations of the chemical shift. On exponentials that stem from these locations, there is a downward shift in the frequency caused by the shielding effects of this tissue. Therefore, the exponential which indicates the effect of this is put inside the construction equation. After adding this effect, the image was reconstructed as in the ideal case. Then, both of these effects were considered in the case-space construction equation at the same time in order to see both of these effects in a single image. In order to examine these effects, a T1-weighted MRI image was acquired from the BrainWeb database. Then, using the parameters that are displayed here, these methods were utilized on the image.
In the results part, we have four main steps. Firstly, the case-space representation of the image is calculated with line-by-line scanning trajectory. Then the trajectory begins from the left below pixel, and the specific two-dimensional complex exponential for each signal is constructed. Then we added all the exponentials to reconstruct the image. Firstly, we observed and analyzed the ideal case by changing resolutions. We observed that 2x2 resolution requires 5 minutes to reconstruct the image, when 1x1 resolution requires 20 minutes for reconstruction process. First observed case-space representation of the image with 1x1 resolution without considering the non-idealities, as you see in the figure. Then we compared the reconstructed images without any non-ideality effect in different resolutions. We observed that while the resolution is decreasing from 3x3 to 1x1, the success of the reconstruction is also increasing. But the duration of the reconstruction process is also increasing because we are sampling more points. We observed that when zero inhomogeneity effect increases, the distortion in the image also increases with varying delta B0 values, which show that the change in B0. And as you see from the figures, when we increase the delta B0, the distortion also increases. We observed that the effect of chemical shift in fatty tissues. We first took shielding constant as 3.35 ppm for the fatty tissues and experienced the effect of chemical shift. Then, in order to examine the chemical shift more noticeably, we exaggerated the shielding constant to 167.5 ppm and observed the result of chemical shift non-ideality in the reconstructed images. In order to choose the white matter in the image, we used region of interest ellipse function of the MATLAB. And you can see the results in these figures. Finally, we added both non-ideality effects of B0 inhomogeneity and chemical shift of the white matter. We took maximum B0 change as 5 ppm, the shielding constant as 3.35 ppm, and the gradient of X as 10 millitesla per meter. As a result, we observed both non-idealities in the same reconstructed image. Then we compared the ideally reconstructed image with the reconstructed image with both non-idealities. Both images were reconstructed with 1x1 resolution. You can see our final results. By changing the resolution of the image, the duration of the reconstruction algorithm and the clarity of the reconstructed image were analyzed in the ideal form. Then we showed the effects of non-idealities both separately and together on the reconstructed images. All in all, one could say that we successfully demonstrated the non-ideality effects in MRI in our MATLAB simulations. In order to improve our results, we can add much more non-ideality and we can use more effective codes to reduce the duration of the reconstruction process. We can also change the inhomogeneous B0 map and we can try different inhomogeneity maps. We can also add the chemical shift effect of the other tissues apart from white matter as further research. Thank you for listening to us, and now I want to talk about our code. In the MATLAB code of this project, we first get the original brain image from BrainWeb website. Then we determine some values related to construction process, like gyromagnetic ratio, field of view, full width half maximum, sampling rate, gradients, maximum B0 inhomogeneity, shielding constant for chemical shift, of course, and Larmor frequency. You can see these parameters here. Then, in the next step, we sampled each predetermined points, and by using for loops, we added each corresponding exponentials for ideal MRI simulator without considering any non-ideality. Then we showed the reconstructed image with 1x1 resolution, and by using region of interest ellipse function of MATLAB, we determined the white matter region for chemical shift. You can see our results here. This is the original brain image. This was our reconstructed image, and you can see the function of region of interest ellipse here. And this is the white matter map. Afterwards, we simulated B0 inhomogeneity as circularly increasing non-ideally. You can see this part. And the result is here. This is the inhomogeneous B0 map. Then we showed the effects of B0 inhomogeneity and chemical shift effect separately. We used the same for loops with additional exponential terms that refers to non-idealities. In order to see the effect of non-idealities clearly, we also exaggerated the non-ideality properties. You can see B0 inhomogeneity here and the chemical shift effect here. these are the results. This is the B0 inhomogeneity effect. And this is the chemical shift effect. In the final step, we show the effects of B0 inhomogeneity and chemical shift of the white matter together, as shielding constant is 3.35 ppm and delta B0 is 5 ppm in these for loops. And you can see our final result here. Thank you for listening to us.